Hello and good morning everyone. Welcome to our paper presentation on comparatively analysis of supervised cell type detection in single cell RNA-seq data. This is a collaborative work with a PhD student, two master students and our professor Dr. Luis Ruda. I am Sheena Hora, hello, a master student from University of Windsor in the School of Computer Science. I would like to start my presentation with an introduction of single cell RNA sequencing data. Today, it is possible to obtain genome-wide transcriptome data from single cell using high-throughput sequencing. This is what we call single-cell RNA sequencing. The figure on the right shows the general overview of single-cell RNA-seq, which consists of different steps. First step is isolating single cell from tissue sample and then sequencing those individual cells. We use single cell RNA-seq to measure gene expression in the cells. Once we have the read count, we compare the gene expression profile of single cells. We compare it using bioinformatics or computational methods. Single cell RNA sequencing helps us to retrieve knowledge about the transcriptomics at the molecular, gene or nucleotide level. High resolution and better understanding of cell's microenvironment is also performed using single-cell RNA-seq data. It also helps to find marker gene, which is one of the major tasks in disease studies. The first single-cell RNA-seq study was published in 2009. Since then, there has been growing interest in doing single-cell RNA-seq studies. The technological advancement of single-cell RNA-seq allow researchers to explore the true diversity of gene expression at the single-cell level, that is masked by traditional bulk RNA-seq. Identifying cell types using gene information is vital and important, which unveils the diversity of cells in the human body. It helps us to give better understanding of a cell's function in its microenvironment. It is also a significant step for studying diseases, and consequently it leads to better diagnosis, drug discovery, and prognosis. Next, for in-depth analysis of single-cell RNA sequencing data, identification of cell type is an essential step. So we have several unsupervised learning methods which have already been developed to identify the cell types. But annotating cluster with the correct cell type required considerable efforts using marker genes. That's why to automate the process of annotation of cluster of cell types, we can take the advantage of classification methods. The classification method use feature selection algorithm to improve the prediction accuracy by finding the most informative features among many in high dimensional dataset. This study also gives the potential use of biomarker genes to improve the automatic identification of cell types. Finally, we biologically validate the cell type marker genes which were extracted from the best feature selection method. Next, coming up to the problem statement, we have a complex high-dimensional single-cell RNA-seq data as an input, and our aim is to predict the cell type in single-cell RNA data in a comparatively supervised analysis. We are doing so by applying three state-of-the-art supervised classification methods, namely support vector machine, SVM, k-nearest neighbor KNN, and random forest RF, with three feature selection methods, namely information gain, ANOVA F value, and chi-squared. Next, this is the pipeline of our method, which consists of four steps: pre-processing, hyperparameter tuning, feature selection, and machine learning model. I will explain about each steps in detail in my upcoming slide. The first step, pre-processing. These steps include basic filtering, normalization, log transformation, and scaling. To perform pre-processing, we followed the standard pre-processing pipeline ScanPy in single-cell RNA-seq data analysis. This step includes filtering unwanted variation. So, low quality cells are represented by a low number of expressed genes. Firstly, we filter out low expressed genes that are detected in less than 3 cells. Next, we investigate the distribution of data which shows low quality cell. Based on the figure, we can see that the number of genes expressed in the count matrix is dense for less than 4000. So we filtered out the cells. So we filtered out these 7 cells to remove the low quality ones. Next, this figure shows the Boolean plot of number of expressed gene in count matrix, total count per cell, and percentage of count in mitochondrial genes. These all help us for the filtering. Next, normalization and log transformation. Normalization is performed to balance the data by bringing it to a common scale without losing any information. It is essential for for reduction of technical noise and bias. The figure shows the effect of normalization before and after on the highly expressed gene. This figure shows the dispersion of genes before normalization and after normalization. Once the data is normalized, the data matrices are log x plus 1 transformed. After per gene quantification, we selected a subset of highly variable genes to use in downstream analysis as they are informative. Genes are extracted based on the mean and dispersion values as mentioned here. The next step in our proposed pipeline is hyperparameter tuning. This step is used to find the best parameter for the classification method. It is done on the pre-processed data. So, to select the best parameters of the classifier KNN, RF, and SVM, we used a Bayesian model-based optimization approach with Gaussian as an adaptive hyperparameter approach. So, this table shows the best parameter found for each method for each dataset. So this, like for example, k equals to 5 is the best parameter for KNN for dataset 1. Now, the next step in our proposed pipeline is feature selection. The most important features were extracted in the feature selection step. The motivation behind feature selection, or we can say gene selection, in cell type identification is that cell types are often distinguished by only few essential genes, which are known as biomarkers. So this is the primary motivation of selecting the features, or we can say genes, in this step. So we use three feature selection method here in our study: ANOVA F value, which is analysis of variance, chi-squared, and information gain. We are using the three classification method in our study, which is KNN, K nearest neighbor, random forest, and support vector machine, SVM. So these three classification methods combined with three feature selection algorithm were evaluated to find the best model. So finally, the cell types are predicted by the method which gives us the highest accuracy. So we are performing our study on three single-cell RNA-seq dataset, which is publicly annotated: data 1, data 2, and data 3. The details of the dataset is depicted in this table, like the name of the dataset, its corresponding tissues, accession number, cell type, cells, and genes. So we are generalizing our experiment on the data set, first two data set, which has same number of genes and same number of cell types. And we use this third data set PBMC, which has more number of genes and number of cell type compared to other two data set. We have selected these data set to generalize our experiments. The dataset is retrieved from NCBI's Gene Expression Omnibus. It contains the gene expression profile of human pancreas and peripheral blood cell lines. Next, this study investigated three general purpose feature selection approaches: ANOVA F value, chi-squared, and information gain, to select the sorted list of genes. So the best number of genes for the training model is chosen by calculating the model performance for top k genes, where k equals to 100, 200, 300, and 400. So we evaluated the accuracy of the method by varying the number of marker genes based on different computational approaches. Here we can see this table shows the classification accuracy with best selected feature for the dataset 1. By seeing all the values, we can say like the information gain achieved the highest accuracy of 98.08 with the 400 number of features. It means like the high accuracy of 98.08% means that the features that have been selected by SVM in information gain are highly correlated and significantly help fulfill our objective. Next, this plot shows the average performance of SVM classifier, which is combined with three feature selection method. This shows the other performance matrices F score, precision, and recall percentage. This figure shows the average performance of these performance matrices for KNN classifier combined with three feature selection methods. Next, this figure shows the average performance of render forest classifier for these performance matrices combined with three feature selection method. When we see the values of all these when we see the values of all these, so we concluded that that SVN combined with IG perform best among all the combination of approaches. Thus we concluded like SVM is able to predict the to predict the cell types, or we can say it achieved the high performance among all other approaches. To biologically validate, we have three steps. Firstly, we selected the top 20 genes which were identified by best feature selection method. Next, we query them into the biological database. Lastly, the cell types with highest overlapping genes are selected. Biologically validate cell type marker genes which were identified by best feature selection method. So we evaluated the performance of our method for detecting the cell type using the high ranked feature or differential express gene through investigating the current literature and reference database. This is what I explained in the previous step. So by investigating GSEA on the result of data 1, which is dataset 1, we found like now 9 out of 20 overlap genes between the pancreas gene set and the top genes which were found by our method. So this table shows the description of the genes, shows the gene names along with its description. Similarly, the this table shows the overlap genes of the dataset 2 with muropancreas ductal cell gene set. These are the gene names and its description for dataset 2. Next, this is the gene information of the overlap genes for the dataset 3. So, in this study, we did the comparatively analysis of supervised identification of cell type using three feature selection method combined with three classification techniques on annotated dataset. We investigated the similarity among features using three state-of-art feature selection method. We highlight the use of only subgroup of highly effective genes, which we also call biomarker genes. These are selected in the feature selection step to find the cell type. Finally, we biologically validated the cell type marker gene which were identified by feature selection step. This is the complete references, and I would like to acknowledge University of Windsor Vector Institute Compute Canada NSRESC. With this, I would like to thank all of you, and also if you have any question or suggestion, please feel to contact us on the mentioned email addresses. Thank you so much for listening to my presentation. Thank you. Have a good day. Bye bye.